All right, good evening, everyone, and welcome to WMRC's Lecture Series. Tonight we will be hosting James Yu, who is a 2023 UC WMRC mini grant recipient and a doctoral candidate, and will be defending his dissertation this summer at UC San Diego in the Biomedical Sciences Department. My name is Galen Kinsey, and I am the scheduling coordinator here at UC White Mountain Research Center. And I'm the one that scheduled these talks that you need to come to. This talk is being recorded and will be posted to our YouTube channel at a later date. And before we get started, I would like to give our land acknowledgement. The White Mountain Research Center acknowledges the Numu, Paiute, and Niue Shoshone as the first peoples and traditional land stewards of Paiahunadu and Coho, the Owens Valley and White Mountains. As a facility managed by a land-grant institution, we pay our respects to the elders, our relatives' relations of past, present, and emerging generations. Thank you. It's all yours now, James.
All right. Good evening, everyone, and thank you, Galen, for the introduction. As Galen mentioned, I'm a doctoral candidate at UC San Diego in the lab of Tatum-Simonson. And today I'm going to talk about a study where we investigate the effects of a multi-strain probiotic. Now, before I get into the specifics of the study, I do want to give a brief background about why I got interested in this field of research in the first place and what our lab generally does. Now, before beginning graduate school at UC San Diego and coming to UC San Diego, where it actually looks like this most of the time, I was in the Army's flight school. And while at the Army's flight school, we learned that we have some regulations regarding flight at high altitude. And so above 10,000 feet, we actually are restricted amount of time that we can spend up there. Above 10,000 feet, we can only spend an hour. Above 12,000 feet, only 30 minutes. And above 14,000 feet, we need to have supplemental oxygen with us. And so imagine my surprise when I came to UC San Diego and I met with Dr. Tatum-Simonson. And she told me about some of these populations that have lived above 10,000 feet for hundreds of generations. And so these are people that exercise, have families, work, all above 10,000 feet. And I thought this was absolutely incredible. I mean, here I am, I'm regulated in how much time I can spend above 10,000 feet piloting a helicopter, but here are people that have lived up there their entire lives. And so this is actually a picture of a Sherpa from last year in the last climbing season at Mount Everest. He actually transported someone that had fallen unconscious for 6 hours with this man on his back, and this is above 26,000 feet. So feats like this are absolutely incredible, and it speaks to the variety of the response to humans to high altitude. And so this is really what drove me to get into this field of research in the first place. And so as humans migrated outside of Africa approximately 55,000 to 65,000 years ago, they encountered multiple different landscapes. And this included high altitude regions such as the Tibetan Plateau, the Andean Altiplano, and the Ethiopian Plateau. And so our lab is interested in how these individuals have adapted to the local environments at high altitude. And so here's a picture on our way to Cerro de Pasco, Peru, where we conduct a lot of fieldwork studies. And here is a picture from when I spent 2.5 months back in Nepal in 2019 conducting some studies in the Himalayas as well. Now, more recently, we've become interested in humans that reside at sea level and that go up to high altitude, because a lot of changes will happen in the body when this occurs. And here's an aerial view of San Diego. Obviously, San Diego is very much the quintessential sea level. And so when humans ascend to high altitude, a lot of different things can happen in their body. When they go from sea level to high altitude, we call it acute hypoxia, because it's a short exposure to high altitude. They haven't been there for a long period of time and had previous acclimatization. And so when this occurs, a lot of different changes throughout the body, and most of them can be negative or have negative symptoms. I'm sure many of you here, if you've gone to high altitude before, you've experienced acute mountain sickness. And in certain cases, it can be more extreme, and it can develop into things such as high altitude pulmonary edema or high altitude cerebral edema. And both of these conditions can be fatal if left untreated. So despite all these potential negative health consequences of high altitude exposure, humans continue to go up to high altitude. Millions of people will either reside at high altitude and work there. This is an example of a mine in Chile where people will go up to these mines, spend a couple weeks there, then come back to their residences, which is a little lower, and then they'll do this periodically. So they'll go up and down for a majority of their life. In addition to this, you have people that love to ski. I'm sure many of you are familiar here, being close to the Sierras. But a lot of people throughout the world will go up to high altitude for a weekend just to go skiing. And some of those individuals will experience symptoms of acute mountain sickness. So with all this in mind, a key question in the field is, are there any interventions that we can utilize to help prevent the development of acute mountain sickness and also improve acclimatization at high altitude? Now, some interventions do exist. I'm sure some of you here are familiar with what's called Diamox, or otherwise known as acetazolamide. It's a prescription drug, so it must be prescribed by a doctor, and it does actually really help people acclimatize to high altitude. However, it does require a prescription, like I mentioned, and also has some side effects that some people do not really enjoy. And so with this in mind, there aren't really many other acclimatization interventions or improvements besides just going slow. And oftentimes people don't want to do this because they want to get up there so they can ski, or they can mountaineer, or they need to work up there. Now, this is where probiotics come in as a potential intervention. So over the last several decades, a lot of research has gone into what's called the gut microbiome and understanding the microbiota that live on and inside our body that we have a commensal relationship with. Probiotics serve as one way to manipulate that microbiota within the gut. And while there has been a lot of hype and a lot of attention to this world, it has really gone somewhat unregulated and unexplored in terms of sound scientific studies. And so recently, last year, we actually had an industry partner. He had a probiotic, which is called Sevomix 800, otherwise colloquially known as SLAB51. And he said, hey, I think that this probiotic can actually improve acclimatization at high altitude. And he based this off of several studies that they had conducted, such as looking at oxygenation during exercise and hypoxia. And so this was done in a hypobaric chamber while someone pedaled on a bicycle and they measured oxygen saturation. Looking at oxygenation during commercial flight. And so in commercial flight, air cabins are usually pressurized to about 8,000 feet. Even something as low as 8,000 feet can have a significant effect on oxygen saturation. And so utilization of the probiotic actually helped improve that. In addition to this, it was also shown to reduce oxidative stress from sleep loss in rats. So when humans, rats, most mammals undergo sleep loss, they actually increase their oxidative stress. And this probiotic was shown to actually reduce that amount of oxidative stress that they experienced. And lastly, it was actually utilized in different clinical studies where they looked at patients with severe COVID-19. This was really during the peak of the COVID-19 pandemic and done in Italy, and it actually improved their oxygen saturation as well. So keeping these preliminary studies in mind, we decided to ask the question, can this probiotic SLAB51 improve acclimatization to high altitude? And so the obvious question now is, what actually is SLAB51? So it's a probiotic, and it contains about 800 billion live bacteria in each sachet. And each sachet is a little envelope that contains the powdered form of the probiotic. And it contains 8 different bacterial strains. This is all available online. You can look up what they are. But this is a commercially available product in Europe, and more recently in South Korea. So you can actually go online and order this if you wanted to. And so, however, it hadn't really been utilized in the context of high altitude acclimatization, and that's exactly what we were interested in doing. So this is what our study design looks like. We plan to conduct a double-blinded, placebo-controlled crossover study with repeat exposure to high altitude in a sea level population. So what this means is that we have a placebo and a probiotic. The placebo is the exact same as a probiotic, just without the bacteria. So it has the same look to it, has the same amount of weight to it, and it's just in a powdered form. And double-blinding just means that the researchers don't know which is the placebo and which is the probiotic, and also the participants in the study also do not know which one's which. And in a crossover study means that we bring them up to high altitude on 2 separate occasions. So our primary outcome from the study was to measure oxygen saturation, which is really the key hallmark of what goes down when you ascend to high altitude and a good predictor of how you will feel at high altitude. Some of these secondary outcomes that we did measure along with this are ventilation, heart rate, sleep, blood pressure, exercise, and cognition. So here is kind of a really broad overview of what the study looks like. First, we conduct a host of measurements at sea level in San Diego prior to ascending to high altitude. Then we bring them up to high altitude for the first time. And then at least 4 weeks later, to get rid of any acclimatization that had already occurred, we bring them up to high altitude another time. Then we compare the 2 different treatments, and at this point, we're still blinded as to which one's which. And then after conducting the statistical analysis, we unblind and we see which one's which.
So how many people did you have participate in?
So I'm about to show that here in a bit. That's an obvious good question, yeah. The plan was to conduct this at Barcroft Station, which is located at about 3,800 meters or 12,470 feet. Barcroft Station is kind of the perfect environment for this. It has places for beds for people to sleep. It has a staff kitchen so they can cook all the meals for you, and it's accessible by car because we had to bring up a lot of equipment with us. And so it's a really unique resource to have here within the UC system, and we're really lucky that we have actually access to a facility like this. And so we brought individuals up from San Diego all the way up to Barcroft Field Station. This can take anywhere from 7 to 12 hours, as we realized, depending on traffic. So we learned that the hard way. And we actually decided to focus on our participant recruitment on the local Latino population. And we chose this for several reasons. One is that they're the largest minority group in the United States, and they're the fastest growing. And despite this, they're often underrepresented in studies. Another reason is because, as I mentioned earlier, we study people of Andean genetic ancestry at high altitude, and a lot of their genetic background actually is similar to Latino individuals. And so we're hoping that one day some of the study results that we have from this can translate over to what we understand about humans that reside at high altitude with an Andean genetic background. Now, in addition to this, an obvious one is that there's a lot of, there's a big local Latino population from San Diego, so it kind of helps to recruit individuals that are willing to go up to high altitude on multiple occasions. Now, we did have some exclusion criteria, which included no recent acclimatization high altitude. So we asked that any of these individuals, we made sure that they did not ascend to above 8,200 feet at least 4 weeks prior to the beginning of the high altitude portion. We are looking for generally healthy individuals, and we did this all through a questionnaire and a screening process. And BMI less than 35, and what's called a stop bang score less than 3. A stop bang score is just a questionnaire that identifies individuals that have a higher susceptibility to obstructive sleep apnea. We just want to make sure that they don't have any sleep disorder breathing, because that will affect how you do at high altitude. Now, with all this in mind, this was our proposed timeline of all this. So this is where we had our proposed sample size. We originally wanted to have 32 people, bring them up to high altitude on 2 separate occasions, as I mentioned. And we would keep them up there for 3 nights and 4 days on each exposure. So we'd have a first group go up and then a second group go up. And we would conduct different measurements throughout the duration of their stay. Now, the best laid plans of scientists and people in general always go awry. So this portion got canceled due to weather. We had the first hurricane to hit California in over a decade, right smack dab in the middle of the first exposure to high altitude. And so this portion also had to get rescheduled due to weather. So in reality, this is what we actually ended up with. Way less people than we wanted. So we had 8 people here, 6 people here, 4 and 12 here. And some of these individuals were repeats, some of them were not. So we actually ended up having 17 unique individuals that went up to high altitude. And based off the limited sample size and some of these individuals not going up twice, we decided to get rid of the crossover component of the study. So this is just going to be a placebo-controlled double-blinded study, not a crossover study. So let's talk about different measurements now. So 3 times a day, we measured their oxygen saturation. This was done via pulse oximeter on the forehead. We measured their resting heart rate and their blood pressure. On a daily basis, we conducted what's called an acute mountainous sickness survey, and I'll get more into that here in a bit. Cognitive tests, sample collection, which included blood, saliva, and fecal samples. Heart rate variability, and I'll explain a little more of that in a bit here, as well as sleep studies. And then 1 to 2 times overall for each exposure to high altitude, we measured their ventilation as well as their exercise. And for the exercise, we quantified how much lactate they produced during the exercise portion. And I'll go more into detail for some of these procedures here in a bit. Now for the actual intervention itself, they either took the placebo or the probiotic up to 3 times a day. So once upon arrival, and we arrived in the evening. And then the 2 full days we spent at high altitude, they took the treatment 3 times a day, usually approximately within 30 minutes after their meals. And then on the last day, they took one in the morning before heading back down to San Diego. So here now I'm going to go into the results of our study. For some of the measurements, I'll go into the methods before the actual results, so it's fresh in your mind while I go into the results. So oxygen saturation, resting heart rate, pretty simple here. We have what's called a pulse oximeter. Most of you are probably familiar with the one that goes on your finger. We utilize one that goes on the forehead because it tends to be more accurate. It reduces the error rate due to any blood perfusion issues or temperature issues. So a forehead pulse oximeter is generally more accurate. And so this is what it looks like here, and it helps quantify oxygen saturation and resting heart rate. And so this is our primary outcome here, and this is what the results look like. You can see on the x-axis here, you have time. So this is all time spent at high altitude. And you can see it goes all the way from day 1 to day 4. And on the y-axis, you have oxygen saturation measured as a percentage. In red, you have the probiotic group, and in blue, you have the placebo group. And you can see right from the outset a clear distinction between the placebo group and the probiotic group, with the probiotic group having a higher oxygen saturation. And it's somewhat maintained through most of the time points spent at high altitude. Now, when we do a statistical analysis, we do a 2A ANOVA, looking to see if treatment or time point have an effect on oxygen saturation. We see that treatment has an effect on saturation, but not time point. And the p-value for that is low, signifying there's probably a difference. And then when we do a one-tailed t-test at each of these different time points, you'll see here the time points are listed out. You can see the associated p-value for each of these time points. Now, about half of these meet the p-value of 0.05. If you're into statistics at all, that number is pretty arbitrary, honestly. But you can see that there's a clear trend here in the days that weren't considered significant. And those days probably weren't significant just due to a limited sample size.
Are the dots individual?
Dots are each individuals, yes. And if you notice here, there's a little more dots in the first half than the second half. That's due to the hurricane. So we had to leave early, so we got a reduced sample size for the second half of the first visit to high altitude. So this is our primary outcome, and we were honestly really surprised by this, really excited. But I mean, we plotted all this even before we knew which one was which. We saw that there was a clear distinction. So before we unblinded the study, we really hoped that the higher one was a probiotic, and it ended up being that way. So going into some of the other measurements, so here's resting heart rate. This is kind of zoomed out to show the averages on each day. And although there's might be a trend here, there's really no significant difference. If you see these bars, those are the standard error bars. And so they overlap significantly, indicating that there's really no detectable difference between the two groups. So resting heart rate is not really changed. And here's that same plot, basically just expanding upon the different time points of each day. And as you can see here, there's really no clear distinction between the two groups. So resting heart rate, as far as we can tell, is not significantly altered due to the probiotic. Now next, we go into what's called the Acute Mountain Sickness Score, otherwise known as AMS. And so this is actually a questionnaire that we ask participants. They just fill it out on an iPad, nice and easy, and it only asks them 4 questions. Actually, this bottom portion here for the sleep question actually got removed back in about 2017 or 2018. It's not part of the questionnaire anymore. But these 4 questions help predict or help quantify how many symptoms you're feeling that are generally due to acute mountain sickness. And so a higher score just generally means that you're feeling a lot worse at high altitude. And so when we look at this over time, so same similar axes as before, you have time on the x-axis, but this time it includes sea level. And on the y-axis, you have the acute mountain sickness score. You can see at sea level, people generally don't feel that bad. Hopefully they don't feel bad at sea level. But on the first and the second day, you see kind of a spike in those symptoms, which is very typical to see in individuals that are not adapted or acclimatized to high altitude. But you can see that the spike here for the placebo group is higher than for the probiotic group. And so on a subjective level, these individuals are reporting that they feel better than the placebo group. Than the placebo group. So a higher score here indicates feeling worse. And then when we do a similar 2A ANOVA here to see if treatment and time point have a significant effect on the AMS score, we see that both of these factors are significantly affecting AMS score. Generally, by the time you're spending day 3 or day 4 at high altitude, people start to feel better because the body is doing what it's supposed to do. It's acclimatizing to high altitude. But it seems like for the probiotic group, you start to feel better faster. Now, next is a sleep study. We utilize a device called the WatchPat, and it's basically a probe that goes on the finger, and that measures oxygen saturation throughout the night. And then you wear a watch, and that measures it collects all that information and then sends it to your phone, so it's all transmitted through Bluetooth and uploaded to a server. And then there's also a microphone that gets taped to the chest as you sleep, so it can detect any snoring that you have as well. And so this is a device utilized to capture sleep disorder breathing or sleep apnea at high altitude. And so here's what that looks like. Once again, similar axes. On the x-axis, you have time, and on the y-axis, you have the average nocturnal saturation. So this is the oxygen saturation average throughout the night while they're sleeping. And once again, I have sea level here just to show that these people generally are mid-90s or higher when they're at sea level. And then when you go to high altitude, you'll see that everyone's saturation decreases, and this is very expected, very normal. But you do see that with the probiotic group and the placebo group, you see a distinction between the two groups. And so similar to before, when we do a 2A NOVA, we see that the treatment has a significant effect on mean nocturnal saturation. So I'm seeing the same direction of effect here as well. Now next, fasted glucose. I'm sure some of you have actually probably done this here before, but it's just a quick prick of the finger. So you get a blood drop, and then there's a glucose strip that quantifies how much glucose you have. And so this is all done in a fasted state. You'll see sea level here. So this is on the morning of going to high altitude. So this is that same day before they made that long drive. And then obviously we don't have a day one, because day one when they arrived at high altitude, it was in the evening and they were not fasted. So you have day 2, 3, and 4. And there's kind of a trend here, but statistically not different really. If anything, you'll see that on day 2, it's probably the closest to being significantly different. But when we do the analyses, there's nothing detectable. The reason why we do fasted glucose in the first place is because disrupted sleep and sleep at high altitude is generally believed to disrupt your body's ability to regulate insulin. So generally, your fasted glucose will actually be higher upon ascent to high altitude as you're acclimatizing to high altitude. So yeah, not significant for the treatment. So next is ventilation, and this one requires a little bit more in-depth explanation of how we collect this data. So when humans ascend to high altitude, they experience that reduction in oxygen saturation, and people will increase their ventilation. And this increase in ventilation in response to lower oxygen or hypoxia is called the hypoxic ventilatory response, the HVR. This is kind of known as the first line of defense, really, against lower amounts of oxygen in the environment. And our lab has a strong history of conducting these measurements. It's somewhat takes a long time of training to be able to conduct these measurements. And so here we are in Salt Lake City doing these measurements in a Tibetan population that resides in that area. And then here it is again in Nepal using a slightly different setup, but same idea. We're quantifying how much breathes in response to low oxygen. Now, how we actually do this, this diagram here made by Eric Heinrich is very handy to show this. We have basically a breathing apparatus here that's one directional. And then, so an individual is connected to this breathing apparatus via this mask. And on this mask, we have O2 and CO2 sensors that can measure how much oxygen and CO2 they're breathing in and out. And this is all through a one-way breathing valve. So air can only come in through here, and when they breathe out, it can only come out through here. Now, this whole system is connected to this tube right here that is connected to what's called a rotometer. A rotometer is just a way to finally control what gases you're feeding into the breathing apparatus. And so this rotometer is connected to different compressed gas tanks. So you have oxygen, nitrogen, CO2. And with these three gases, you can actually, on a really precise level, control how much of gases they're breathing in. And you can manipulate what they're breathing in. And then over here, you have what's called a pneumotachograph. And that actually will quantify the volume of air that you're breathing over time. And so you can get how much someone actually breathes in and out over time. And so here's what it looks like up at Barcroft Station. You have the breathing apparatus here. Over here, you can kind of make out the little tube back here, and that is connected to a rotometer that's outside the picture right here. That's connected to gas tanks that are further out to the left. But this is what it looks like. It can be a little scary at first when someone is unfamiliar with it, but it's totally safe. We've done this in many different parts of the world now. And all of this data gets fed live to these sensors, which are then input into this computer where we're watching in real time all the changes physiologically that are happening.
And how long is that procedure?
So that's a perfect question. So here's our breathing protocol. So this is a little busy of a figure here, but on the x-axis you have time. So overall it takes about 35 minutes for the actual breathing portion. On the On the y-axis on the left side, you have the partial pressure of oxygen, and on the y-axis on the right side, you have partial pressure of CO2. So the first 5 minutes right here, they start off, they're just breathing room air. So room air is literally just the air of the room. This valve right here can be turned to open to the general ambient air, and when that's open, that's all they're doing. And that's just to get them, acclimatized to the breathing apparatus to make sure they're not trying to control their breathing. Because some people do get nervous, but after 5 minutes, usually people just calm down and really, we're just trying to keep them awake. That's probably the hardest part of all this. So after 5 minutes, we start the first stage, which is called the hyperoxia stage. And this is where we actually have a higher level of oxygen. Typically, sitting at sea level, we're at about 20.95% oxygen. In the hyperoxia stage, we give them 30% oxygen. And this is kind of getting into the nitty-gritty of the basic science of ventilation, but it's to get rid of any ventilatory acclimatization to hypoxia that a person already has. So for example, if you're at high altitude, as soon as you start getting into lower levels of oxygen, you're already starting that process to acclimatize and ventilate different at high altitude. And so this process actually gets rid of that at first because we want to see what your baseline is. So that's the first 10 minutes right there. And then in the 5 minutes after that, we call what's normoxia. And so normoxia is just what you would experience at sea level. And so we give them 20.95% oxygen. And then after that, we start the first hypoxia stage. So while they're here in normoxia, we determine what their natural end tidal CO2 is. So we haven't changed the CO2 here at all yet. And it's just what their natural CO2 baseline is in their blood. And we measure that via, at the end of the breath, right at the end is the amount of CO2 due to perfusion of the lungs and the ambient air is the actual CO2 that's within your blood. So it's a good proxy measurement for that. And this is really important because the body responds by ventilating more in response to lower oxygen, but it also responds separately to increased amounts of CO2. There are two separate feedback loops to the brain to breathe more, and they actually act independently and synergistically as well. So it's really important when you're trying to quantify how much a person breathes in response to low oxygen to control for the CO2 if you're only interested in the oxygen portion. So that's exactly what we do here. It's called an isocapnic hypoxia maneuver. And so we lower the amount of oxygen that they breathe while keeping the CO2 constant. And then right after that, we go into 5 minutes of what's called hypercapnia. And so we bring up the oxygen back to the normal levels that you see at sea level, but then we increase the CO2 by about 5 millimeters of mercury. So this way we're isolating the ventilatory response to just increased CO2.
So,
Yep.
the CO2 is an input?
Yes. So yeah, going back to
And
here.
why is it negative?
So it's, this is just in relation, it's on this axis right here. So it's actually not negative. Yeah. It's a little bit of confusing setup here of like, yeah, which one's which. But we generally will just increase 5 millimeters of mercury of CO2. Which for most people is really, you can't really even feel that. Yeah. And then lastly, we do a combined lower oxygen and increased CO2. And so we see the additive effect of both of these components that increase your ventilation. So that's the entire protocol. Here I'm going to show you the results of that first stage of hypoxia in these next figures. So here is the ventilation in response to lowered oxygen. And so once again, you have time point on the x-axis, and you have the ventilatory response on the y-axis. So you have sea level here. You don't really see a difference between the two groups, but as you spend days 2 and 3 at high altitude, you see a clear distinction between the probiotic group and the placebo group. So it seems like these individuals are breathing more in response to lower oxygen.
When you say breathing more, is that just volume?
Yeah, so the HVR itself, so it's volume over time, and then it's corrected for the amount of oxygen saturation that we decrease them by. Because if you bring someone down by 10%, it's going to be less than if you bring them down by 20%. Because obviously the deeper down you bring their saturation, the more they're going to breathe in response to that. So we have to control for that. And in addition to that, we also control for their body weight, because individuals that are bigger tend to just breathe more because physiologically they have bigger lungs. So this is the results we see, which is really surprising for us actually. And then we see basically a significant difference for the HVR dependent on treatment. Now, I'm going to show you the hypercapnic results. So this is just ventilation in response to increased amounts of CO2. And then you see kind of a similar trend, but not significantly different here actually. And you see here that while the treatment here is not significant, the time point actually is, because generally people's ventilator response to increased CO2 goes up over time as they spend more time at high altitude. And this isn't as well studied, but it's most likely in response to how people breathe in response to CO2 during sleep, actually. Because central sleep apnea is something you can develop at high altitude, and that's actually more dependent on CO2 levels than O2 levels. And then lastly, it's the combined hypoxia and the hypercapnia. And here you do see a clear difference between the two groups here. So this is the synergistic effect of both the lower oxygen and the increased amounts of CO2. And when we do the statistical analysis, we see that the treatment groups are significantly different in their responses. So moving on to heart rate variability. And so heart rate variability is quantifying the amount of variability in the time between heartbeats here. So if you look on this example right here, you see that it's a similar time span in between these beats, and that simulates into a lower heart rate variability, or HRV. If you have more variability in between these heartbeats, it's actually a higher HRV. And a higher HRV is actually generally seen as better. So more variability is actually seen as better. If you guys wear fitness watches or wearables, the newer ones will generally track heart rate variability as well, and they'll tell you, hey, your heart rate variability is going low. Maybe you should improve on your, whatever you're doing, you should probably stop doing. So, so the lower heart rate variability is typically associated with what's called an increase in sympathetic activation. Sympathetic activation is basically your body's your fight or flight response. So you're being activated to be more anxious, more nervous. That's generally associated with having a lower HRV. Now, this is the way we actually quantify it. So we have electrodes that go on each wrist and one on the ankle to ground the signal. And then this all feeds into an EKG that feeds into our computer. So we're seeing this in real time here. And all these measurements were done in fasted individuals. So even at sea level and at high altitude, it was done before they ever ate any food at those instances, because eating can obviously have an effect on your body's sympathetic and parasympathetic nervous system. And so we want to control for that. So this is what's typically seen as the HRV here. And you can see that you see a general decrease at high altitude, which is pretty expected. Your body is under a great amount of physiological stress, and so you activate your sympathetic nervous system a lot more. We don't really see a significant difference here. We do see somewhat of a trend on day one, but not significantly different there. Now on this figure right here, this is more of a measure of your parasympathetic nervous system. Your parasympathetic nervous system is one that actually calms down your body. So think about it as kind of the opposite of sympathetic activation. So this is what happens when, for example, if you eat too big of a meal and you feel sleepy afterwards, you can thank your parasympathetic nervous system for that. So you can see here that it also goes down, which indicates that your body is also not resting as well when you go to high altitude. But once again, we don't really see a significant difference here between the two groups. So in general, based off this, we can conclude that we see an increase in sympathetic activation at high altitude, but really no difference between the treatment groups. Now next is blood pressure. This is done with a typical blood pressure cuff. We would measure them 3 times a day, and each of those times we also measured 3 times, so really 9 times a day, and then we would average all those measurements together. We don't really see a difference here actually. So no difference between the groups, and no real change at high altitude. And this is also pretty well characterized in the literature. Some previous studies have identified an increase in blood pressure when you go to high altitude, but most studies indicate there's not really a big difference or change. So next is exercise. And this was done on what are called cycle ergometers. And so ergometers are basically stationary bicycles where you can control the amount of resistance to indicate a specific amount of workload. So you can control the wattage on the actual bike itself to adjust for things like body weight. And so for the exercise, we measured their lactate levels before and after they bicycled for 10 minutes in what's called a submaximal exercise test. And so this is not meant to be, a really strenuous task. It's meant to be a submaximal, or you can have a conversation the whole time. And then the way we controlled for weight is we matched how many kilograms they weighed to their wattage. So if they weighed 50 kilograms, then they had a 50 wattage resistance, basically. So, and then we use a lactate analyzer, so it's once again similar to the glucose monitor. It's a finger prick, and then you just put a strip on there, and it'll measure the lactate. We did it before and after they bike for 10 minutes. Now, here is their oxygen saturation on the y-axis, and then on the x-axis, you see this is their what we call their baseline at high altitude. So we would measure their oxygen saturation for 30 seconds before they actually started bicycling. And then we average the last 30 seconds of the 10 minutes of bicycling. So this is just to do kind of a sanity check that we do indeed see a decrease in saturation as they do exercise at high altitude. So for this one person that went higher, I'm not sure what's going on with that person, but the human body is notoriously variable. Maybe this person just always feels better when they exercise. And then, so here's the actual lactate levels. And so here on the y-axis, we have what's called the lactate delta. And so this is the difference of the delta lactates. So because in order to know the difference in lactate, we do measure before and after, but we also need something to compare it to. So we compare it to what they did at sea level. So same exact wattage, same exact amount of work, same exact 10 minutes. They do it at sea level, and then they do it at high altitude, and this is comparing the lactate production of each of those. And you can see it's what you expected. Everyone produces more lactate when they go to high altitude. And this is expected because you reach your aerobic threshold faster at high altitude because there's less oxygen available. And so this is part of the reason why exercise at high altitude is so much harder than doing it at sea level. And so here is what the differences look like when you break it down by the treatment groups. And so this is actually kind of flipped backwards with the colors, but here is the placebo group, and here's the probiotic group now. This is before we unblinded the results. And you see that on average, the probiotic group actually produced less lactate after conducting the exercise protocol than the placebo group. Now, one key thing to note here is that if you look at the previous slide, there are some less data points here. We did remove 3 outliers. If you do include those 3 outliers, then the significant difference does not exist. But we removed some of those outliers because 2 of them conducted exercise on different days at high altitude because they had to leave early. And that will affect the results because all of this was conducted on day 4, so when they're the most acclimatized to high altitude. And those individuals did exercise on day 2. So they're really not comparable. And one of the other people actually fainted, not during the protocol, but prior to the protocol. And so we probably were like, we shouldn't include that person. So I would take these results with a grain of salt, though, just because the sample size is really low. We did remove some outliers, and it is very preliminary. And yeah, when you do a t-test to compare these two, you'll see it's a significant difference. So some of the other results that we have pending that are currently being analyzed by the UC Riverside team are some cognitive tests. So these individuals actually completed a cognitive test at sea level and also at high altitude on every day while they're at high altitude. And so we're really curious to see the results of this. And also, that's one of the other groups at UC Riverside are looking at immune function. So we did take blood, before, after, during all of this, and they're actually categorizing the amount of immune function changes that are happening throughout the blood. And they're seeing some really interesting changes, so we're really excited for those results as well.
Is the white blood cell count?
Yeah. So this is kind of a newer topic in the high altitude world. There's a lot of physiological crosstalk between the immune response and the response to low oxygen. And so some of the similar biological pathways get activated. And so you're actually seeing an increase in some immune cell types and a decrease in others. But generally, you're seeing a kind of a increase in immune response when you go to high altitude. And some of these changes can actually persist for a pretty long time. So that's kind of the most surprising result, actually. So some things that are pending analysis right now, we collect a lot of different samples. So we're looking at the DNA, the RNA, different protein levels and metabolites within the blood. So those are all pending right now. We obviously have the fecal samples, so we're curious to see if any of that is changing with the probiotic usage. We have saliva samples where we can measure biomarkers from. And then, so in summary here, from our study of 17 people, we see significant differences in daytime saturation and nighttime, differences in their acute mountainous sickness score, their ventilation, and how much lactate they produce during exercise. We don't see any significant differences in their resting heart rate, their fasting glucose levels, the heart rate variability, and their blood pressure. And so the big question obviously is what's next? We're really interested in potential mechanisms. This study was, I should probably mention this at the beginning, this study was mostly funded by the creator of the probiotic and the owner of that company. So that's a big caveat here. But he did not dictate any of the scientific results. The science is what the science is. And so he just wanted to say, hey, I'll fund you guys a study and then publish whatever you guys find. And so us as scientists, we're really interested in what is actually happening here. People in industry don't really care. There's like, will it make me money? That's all they care about. We're like, what's the science behind this? So, and one thing that I'm really curious about in the lab is in general is if we see the same effects that we see in the sea level populations at high altitude. So in populations that reside at high altitude that live there, will we see the same exact results? And so with that, I would really like to thank the White Mountain Research Center for, one, having Barcroft Station as a resource available to us, because I don't know where else we would do this kind of study. And really, it was part of the staff and the mini grant that helped make a lot of this possible. Other than that, the research participants, they had a smile most of the time, even though they did not feel very well at high altitude. And so they were really great to work with. I've never heard a complaint from any one of them, so that's pretty fantastic. In addition to that, we have a lot of collaborators, both within the lab and at UC San Diego and at other universities, such as UC Riverside. And so I want to thank all those individuals for making this study possible. And thank you for your attention.
Are there any theories on the mechanism? This seems
Yeah.
weird or profound.
So I knew this question would come up. So here is the proposed mechanism from the creator of the probiotic. So
Sorry, that's really bright.
that's fine. So generally, what they believe is happening is that you consume the probiotic, and in about 2 to 3 hours, it gets to the intestinal level, and that causes the intestinal tract, the upper intestinal tract, to vasoconstrict. So when it constricts, it utilizes less oxygen, and that frees up oxygen for the rest of the body to use. That is their theory. Me as a scientist, I will believe this once I see scientific studies that support this.
So it's kind of like not so much blood and oxygen are being dedicated to like digestion and are free to
Not necessarily digestion, but
intestine.
yeah. So I mean, every organ utilizes oxygen to a differing degree, and the intestinal tract is really large and uses a lot of that oxygen. And so if you theoretically decrease that oxygen's demand on that system, then it should free up oxygen to the rest of the body.
That's
Yeah.
It's kind of like a short-term effect, or what about long?
Well, that's the big question here is if it's short term and if you are actually colonizing the upper intestinal tract, is this something that actually changes to this more oxygen efficient method for a prolonged period of time? Does it last a long time? We really don't know the answer to this. I mean, if you look at pulse oximetry data, which is what we have, that measures the amount of oxygen that's binded to hemoglobin within the blood. And so that's ultimately where we're seeing a difference. And so a next logical step, in my opinion, would be to do a blood gas analysis. And so to actually look at the binding affinity of hemoglobin to oxygen within the blood and see if that's being altered based off the probiotic ingestion. And then obviously there's the ventilation aspect as well. Is this increasing ventilation, or did we just happen to choose people that had a greater ventilation? That we're actually starting to parse apart now. I'm doing a statistical modeling for those questions, and I literally did this yesterday, so I didn't have time to put it on here, but what we're seeing is that both ventilation and ingestion of the probiotic are positively affecting oxygen saturation, but independently. So they are both contributing factors when you include them in a statistical model together, but they each have their own contribution basically. And it seems like the probiotic is not actually affecting ventilation, it's affecting something else that's increasing the saturation.
Then why is the ventilation, or any idea why the ventilation is increasing?
I think that was just by chance. I think somehow we selected, this is all randomized, and we just had a group. When your sample size is only 17 people, you run the chance of that happening, and I think for some reason we just had a group that tended to breathe more than the other. Yeah. Or if there is actually an effect of, it gets complicated because the probiotic could be affecting something within the blood, and effects in the blood can also change ventilation. Because when it comes down to how ventilation works, it's a specific organ within the brainstem called the carotid body, and it's effectively tasting your blood repeatedly to determine how much you should be breathing. And that's actually how it detects, hey, I'm tasting lower oxygen, I need to breathe more. So if you're changing something within blood, it could be changing something in ventilation. So it's all connected, and it's really difficult to parse apart. Yes?
Thank you for the presentation.
Mm-hmm.
One thing I found interesting in your results is that you showed several indicators that the biggest difference was really on the first couple of days at high altitude, and as you got day 3 and 4, the two groups became more similar. And then you also had a bunch of data showing that the two groups diverged more and more day 3 and 4. I was just wondering if you'd comment on that contrast.
Yes, that's a great question. Typically, we see for certain measurements, the greatest amount of divergence on the front end, and then some other ones on the back end. So I think it depends on what measurements you're looking at, but things like acute mountain sickness and that physiological stress is obviously greatest at the first half, because that's when your body just hasn't had time to acclimatize yet. So I think for some of the other measurements where we see less of a difference as it gets later on, that's because the body is compensating the way it should be. It's physiologically acclimatizing to that low oxygen level.
Right, but it seems interesting that the group getting the probiotic treatment is diverging more over time. We're seeing actually a bigger difference.
So when you look at things like ventilation, for example, and I think that's where the biggest divergences occur, that is most likely due, now this is where I really don't know, this goes back to the question is if the probiotic is affecting ventilation or if it's affecting something within the blood that may also be affecting ventilation. We don't know, actually. Typically for ventilation, in terms of acclimatizing to low oxygen, you see the acclimatization process happen within 24 to 36 hours, but it really varies by the individual. So when we go back to those results, it seems like some people in the placebo group are catching up with their ventilation later on, but the probiotic group just tends to have a quicker response in that first full day at high altitude. And so we don't know.
Question. It'd be curious if you would extend the study, say like to 8 days or something like that, to see, like, to expand on this question. Because, you know, I've lived and worked up at Barcroft for many, many years, and I know that the first few days of being up there are the hardest days. So just with what you have here, what I notice when people come up there, if they get sick in the first 24 to 48 hours, there's a chance that they're just gonna go downhill, and we gotta send them back.
Mm-hmm.
You know, and if you can reduce that in the first 24 to 48 hours, it's amazing. But then as you acclimate, like you said, naturally, everyone does. Be curious to see that, like, after, say, 8 days with this probiotic, you know, what it does at that point. You know?
Yeah.
Do you have any plans of doing anything like that?
We would love to. We're really curious on that as well. I mean, we always say in our lab, the first 2 days and the first night is the most telling, because after the first night, you generally will know how someone is going to feel for the next 4 days.
Yeah, and if you could prevent that in the first
Yeah.
24 hours with this, it's
And-
fantastic, because
yeah.
diatox, like you said, is awful.
Yeah.
It's an awful drug, and, well, it has its effects, but, the second question I had was that, with this, I'm sure just because it's a probiotic, there's no effects that you can tell by the blind study, by them taking it, they don't feel anything different, correct? They feel.
Yeah, from as far as we could tell, they didn't know which one they were taking. Yeah, and they didn't know. I mean, you can look at things like the Acute Mountain Sickness Survey, which is a subjective quantification of how someone feels in response to just things that are strictly related to altitude illness. And they subjectively feel better on the probiotic, but in terms of side effects of the actual ingestion of the probiotic, we didn't see anything. But once again, it's really hard to parse that out, because a lot of people just feel nauseous and they get general bloating and gassy at high altitude. And so, I mean, how do you, is it the probiotic or is it going to high altitude? My guess is it's probably just going to high altitude. But I mean, we would love to extend this out to further. We would love to increase the sample size. And we would love to do you know, for the purposes of this study, we really looked at it as an intervention. And so we just had them take the probiotic as soon as they basically arrived at high altitude. I'd be curious to see if they could take it, you know, after a night at high altitude, and if they feel better within a couple of hours, as an intervention that occurs later, where you can identify the people that already feel bad and if they'll feel better, versus someone you don't know if they're going to feel bad or not, and you just give it to them anyway. So there's two different use cases here for that, I think.
I think this product would have huge potential for use in the I feel
Yeah.
horrid at Barcroft. I'm fine at Crooked Creek, and I
Yeah.
from here. So that would like be beneficial just for like our personnel.
I totally
Yeah.
agree. Yeah, I mean, when we see you guys up there, and some of the staff have been up there for like weeks already, we're like, man, that's impressive, because I know it can put a huge strain on the body. And you see the effects on sleep. You see the effects on on the sympathetic activity. Like that's all very real and known. And so there's the markets for this. Coming at this from an industry perspective, there's obviously mountaineers, people that work at high altitude. All those would greatly benefit if this study gets replicated and vastly expanded to include more people. And so I think that's really part of the goal of the funder of the study, is he wants to kind of expand into that market potentially and utilize this as a jumping ground to do more studies.
I'm just curious as to, for the Peruvian people,
Mm-hmm.
been to the Atacama, and they recommended we use coca leaves
Oh yeah,
for the altitude sickness.
yeah.
So, just wondering, like, do they already have like a natural probiotic gut in them that just makes them susceptible to that, or like, how does that really work with people that maybe don't have it or have a good
Mm-hmm.
gut probiotic? Would you even recommend them taking it?
So that is really difficult to parse out once again, and it's really just understudied. I mean, some individuals do really well at high altitude, others don't. You can be an Olympic level athlete and just do terrible at 10,000 feet, and I've seen it time and time again. And that is the reason why I got interested in this in the first place. And when you look at people that resided at high altitude for hundreds of generations, you see that variation once again. People have this kind of idea that, they've lived there, their grandparents have lived there, their ancestors have lived there. Some people in the Andes really do suffer from high altitude illness. They develop what's called chronic mountain sickness, for example, and that's one of the areas that we study actually. Yet some people are totally healthy. And so is it something about their gut microbiome that is affecting their ability to adapt to high altitude environments, or is it something that they're taking, like coca leaves, for example? That is basically the whole reason why our lab does what we do. We're interested in this human variation, and by understanding why some people do better or do worse, we're hoping to be able to apply that to residents that reside at sea level that have diseases related to oxygen as well. So it gets out to the, when we look at the extreme ends of the spectrum in terms of the human response, we understand better the biological mechanisms, and that can be applied to interventions such as this.
So on their product's website, it says take 14 sachets once a day for 14 days, I think it's what
Yep.
it is.
Mm-hmm.
And did you say you gave it to them 3 times a day?
Yeah, we did. Yeah.
So is that just on the first day then?
No, so on the first day they arrive in the evening, so they only take one on that first night, but they spend 2 full days at high altitude. On each of those full days, they take it 3 times a day, generally after each meal. And so this dosage is higher. It's definitely higher than like what a person would typically take this for recreational use. And we wanted to hedge our bets to see, okay, if this is going to have a difference, then we want to make sure we can actually detect that significant difference. So
So I was wondering if you were taking it for 12 days in advance to going someplace,
Mhm.
do you think that would build it up, and would it create a colony that survives or something a little bit better?
that's exactly what we're wondering. We don't know.
So would you recommend, if somebody wanted to try it, would you recommend doing it your way, like after?
If you have unlimited funds and you're willing to experiment with something like this, I recommend taking it beforehand, a couple days beforehand. But that is my unprofessional advice.
The hard part of
Yeah.
this, it is. Is that the only source? I see it's like 96 euros or something like that.
So that is the European website for it. Ormenes is the company for that. But they also, this is very recent, they started selling it in airports in South Korea. It's called O2 Booster, which is a little more of a straightforward name, I guess, than SLAB51 or SIFO Mix 800. So it's commercially available, and I'm guessing that he would like to break into the U.S. market as well. But one important thing to note about probiotics is that it's kind of the Wild West out there right now in terms of regulation. So the FDA doesn't regulate things or what people call what do they call it? Dietary supplements. And so this is typically how probiotics get branded. as soon as you say that this treats an illness or a disease or it's an intervention in terms of a clinical meaning, then the FDA has to step in and you got to go through a full-blown clinical trial to help develop and go through all the phases. But right now, it's a probiotic world out there. It's people just can kind of claim whatever they want. So the creator of this probiotic, obviously, he utilized this in clinical trials in COVID-19 in Italy. So I think he wants to actually build a more robust scientific basis for this.
Any known adverse side effects?
Not that we know of. Just, I think when we filed to conduct this study, obviously anything that involves human subjects, you got to put through an institutional review board. Main side effects were just typical things like bloating, gas, digestion issues. Yeah. And I mean, like I said before, you see all that at high altitude anyway, so yeah.
So clearly this is a proprietary product. Is there anything that you notice that's extra special that's in there? Or is this kind of like garden variety probiotics?
I don't know. I was very skeptical when we were first approached about doing the study, but when someone says I'll fund a research study, you're just like, great, I'll go do that research study. Because, outside of the probiotic aspect of the study, we still learned a lot more about, just the basic physiology of individuals acclimatized in high altitude. So I don't know, what goes into making a probiotic, but based off the results that we saw that I was very surprised by, I think there's something in it that is actually having that significant effect.
So you don't know anything about, you mentioned that there's 8 different species?
Mm-hmm. Yeah, the different strains, yeah.
So you don't know, are those different than what's typically seen in the Westerners?
Mm-hmm. So I know that some of those strains are also used in other probiotics that are out there in the markets, like lactobacilli is one of the first strains that are listed out if you go on the website for it. And I know that is like the go-to for most probiotics that are on the markets these days. But the rest of, he lists out those strains, but in terms of what else he does to that mixture is all proprietary knowledge. So it's he has a patent for it, and so we don't exactly know. But I'm sure, if there are microbiologists out there, they could probably mix those 8 strains together, play around with it a bit, and see if it gets the same effects. But I don't know who's willing to do that. Okay.
All right, thank you.
Thank you so much, everyone.